Now onto one of my favorite topics, enzymes. So this is really nitty-gritty sort of stuff. So part 1, we're going to talk about the diversity of function of enzymes. So we're going to classify enzymes into one of 6 general categories based on function. So as you all know, enzymes are one part of proteins. So if this circle represents all proteins in the world, enzymes are just a small subset of that. So as you can see, all enzymes are proteins, but not all proteins are enzymes. For example, these proteins over here can be used for structure, the proteins over here can be used for cell signaling, that sort of thing. Okay. And so like I said, all enzymes are proteins, so proteins are all of these. All of the enzymes are in this yellow circle, right? But not all of these proteins are enzymes. So keep that in mind. When protein and enzyme are not interchangeable. The enzymes are the specific proteins that do catalysis. Enzymes are for catalysis. These are catalysts. Okay. And so because there are so many, like a ridiculous number of reactions catalyzed by enzymes, we've been able to, after much study and much work, break it down into 6 general categories. And you should be definitely familiar with these categories as we move on with this class, and if I show you a chemical reaction, you should be able to tell me what class it would go to. So number 1, Oxydo Reductases. These are responsible for redox reactions. So that makes sense. Oxido reductases, oxidation, reduction, these are the redox reactions. So these would be like the dehydrogenases. 2, Transferases. So what transferases do is that these transfer functional groups. Functional groups from one molecule to another. Makes sense, right? What color is it using? Here we go. 3, Hydrolysases. These, as the name implies, use water to cut bonds. That is also known as hydrolytic cleavage. So hydro meaning water, lytic meaning to cut, cleavage, in case you were forgetting that lytic means to cut. 4, the ligases. So ligases is a really sort of broad general category. So these are anything that is added across or creates a double bond. So if double bonds are involved, it's a ligase. This is also sort of anything that could be rearranged, so like rearrangements. Around a double bond that would also be ligases. Oh no. Okay, much better. Ligases. Okay. The isomerases, let's get the right color here. 5. Isomerases, these catalyze intramolecular rearrangement. Intramolecular rearrangement. So we're going to see examples of all of these when we get into glycolysis. And finally, ligases. So the ligases, these are joining 2 molecules. These are very important. Joins 2 molecules, and although you would expect this to decrease the entropy, which it does, joining 2 molecules together, 2 is more disordered than 1, this is still a favored reaction. Okay. So we're going to go over examples of these during class.